Okay, hello everyone. Today we're going to talk about this new paper that came out that I've made a few videos on, and I'm hoping that this longer YouTube video can help answer some further questions, help us all understand this research a little better, and continue the conversation, just to make sure there's no misinformation being spread and that we all understand what this paper is trying to say to the best of our abilities. Okay, so the paper is titled Cannabinoids Block Cellular Entry of SARS-CoV-2 in the Emergent Variants. First, I want to show you how to access this publication if you're interested in reading it. So first, you could just Google the name of this paper, and you can do this with any paper. Not every paper is going to be open access, meaning not everyone's going to be able to read all of every paper that you search, because sometimes they're behind a paywall, so you'd have to pay more. But in this case, it's open access, so we're all able to read it, which is awesome. We love open access publications. So if you type this in to Google, something's going to come up on PubMed or say NCBI. That's the one you want to click. You likely won't be able to read the whole paper here. It's just going to bring you to a page that looks like this. So then to actually access the paper, we want to click that link in the upper right corner that says full text links from ACS Publications. And this is because this paper was published in the Journal of Natural Products, which is a great journal for this publication. I'm a natural product chemist, so it's a very important journal in our field. So we click ACS Publications, and then that's going to bring us to this different looking page, but still has the title of the publication at the top. If you want to download the PDF, you click what's circled in the red circle right here. It says PDF, and then you could mark it up by like highlighting it and putting notes in and then saving it as a separate file. So then you can download that to your computer to read it and save it for later. You can also access the supporting information here. So the supporting information is any data that is important to understanding this publication, but it is they don't want to put it in the main text, so they add it as just supplementing information. So it's things like figures, data from some of the assays that they ran, things like that. Some of the tests, assay tests, same thing. Okay. I will put a disclaimer in this, and I'm going to put something at the end too. This is not my scientific study. I'm just relaying the information from the study from the way I understand the authors to be presenting that data. The reason I kind of want to stop talking about this study that much, because I didn't publish this. So this will probably be the last video that I talk about it. It's really, 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 really important to note that all this data that is acquired in the study was done in a laboratory. It's done in test tubes and petri dishes, super isolated systems. It does not represent the complexity that the human body is or that the COVID-19 virus is. So when we look at this data, we need to understand that this might not be translatable to animals or even to humans. We don't know yet. This is just step one. Some of the data has also been generated on a computer. So I'll walk you through that portion of the study as well. And then lastly, this information is just a piece of evidence. It's just a piece of the puzzle. It's suggesting that some compounds might help against COVID infections. This is by no means the entire story or a definitive evidence, okay? It's just evidence that does make sense towards this story. So the purpose of the study that the researchers are proposing here is to search for compounds from the hemp plant that can bind to a portion of the COVID spike protein. In theory, if these compounds that they're testing, and a compound is a molecule, like an isolated molecule, if they can bind to a portion of the spike protein on that COVID virus, the authors are thinking if the compound can bind to that spike protein, it will no longer allow that spike protein from interacting with our cells and then entering our cells and infecting us. So we're kind of just messing up the way that virus talks to the cells. We're like physically getting in the way of those two interacting. So in theory, it would prevent the infection. So first, this is something I really, really liked about this publication, and it's how they found out which compounds were active against the SARS-CoV-2 virus. So I'm very biased here, but I think all studies, especially natural product studies, should incorporate some component of screening. That means that you don't have this bias that a certain molecule is definitely going to work against COVID. Instead, you have this library of different compounds, a whole bunch of different compounds that are different in structure and function. And you screen them using a test to see if they're active against something. So these researchers were using purified compounds from the hemp cannabis plant. So you can think of things like CBN, CBC, and then CBD, CBDA, which we're going to go through what all of these mean, but a bunch of different compounds, not just one single one. Okay, so I want to bring up this is just a portion of a figure that was shared in this publication, but I think it's the coolest. This is what really made me, at first, when I saw this publication, I'm like, I'm not going to like this. I don't like reading things about COVID having, like, or hemp cannabis having these magical powers against COVID. But when I saw this figure, I was like, wait a second, this is really cool. So, essentially, if we're looking at the upper left portion here, it says this is the SARS virus. And these small spike proteins here have different components of the spike protein. So we could call this like I guess this might be the lowest. So upper, middle, lower. Okay. So in theory, the upper, middle, and lower could be targets for preventing the virus from getting into our cells. These authors were using the S1 subunit as their target for having the hemp compounds interact with this unit, and that would not allow the COVID virus from interacting with our cells. So they're essentially looking for compounds from the hemp plant that can bind to this. That is the goal. We're looking for compounds from the hemp cannabis plant that can directly bind to this S1 subunit of the COVID spike protein. Okay? So the first thing that they're going to do is they take the S1 subunit of the COVID virus, and it doesn't actually need to be acquired from the virus. You can actually produce this in the laboratory. It's just the sequence that corresponds to the S1 subunit. So they took that S1 subunit and they immobilized it onto a magnetic bead. So they essentially took that subunit and literally stuck it onto a magnetic bead. So now it's just a bead with a subunit floating around in what we call a 96 well plate. And that's what this is here, the square thing. So each of these things on this well plate is like a little well that holds a little bit of liquid. And so they're going to put the S1 subunit into all of these wells, and then they're going to test different compounds in each well to see which ones are active. So the first thing they're going to do is they're going to put that S1 subunit that's attached to the magnetic bead into these wells. Then they're going to put their compounds of interest. And it's actually really cool that these researchers weren't just testing single pure compounds, they were also testing some extracts and some combinations of different compounds from the hemp cannabis plant. So, in theory, again, we're looking for compounds from the hemp cannabis plant that can stick to this S1 subunit. So the S1 subunit's floating around in one of these little wells. And if something from the hemp plant can stick to that S1 subunit that's attached to that magnetic bead, it's going to all stick together and form this little glob. So that's the S1 subunit, the magnetic bead, and the molecule from the hemp cannabis plant. And then they're forming like a little globular thing together. Things from the hemp cannabis plant that they're testing that are not active against this test to see if it can help prevent the COVID-19 virus from entering your cell are just going to keep floating around. They're not going to bind to the S1 subunit. They're not going to bind to the magnetic bead. They're just going to be floating around by themselves. So then the researchers wash away the things that are not stuck together because they're not on a magnetic bead. So you can put in a magnet, get everything to stick to it that was stuck to that magnetic bead, meaning it was also stuck to S1 subunit, and then wash away everything else. So everything that's not active is just going to wash away. Then on your magnet, you're going to have the magnetic bead, the S1 subunit, and your active compound. Then they introduce a certain solvent or a liquid that's going to wash away the active compound from the S1 subunit. Okay. And that's this step here. So that's what we call a ligand. It's something that was able to bind to something else. So the active compound from the hemp cannabis plant that was able to stick to the S1 subunit is called the ligand, and it's our active compound. But now we're releasing that from the S1 subunit. Why are we releasing that? We're releasing it so we can figure out what it was. And then we use a tool called mass spectrometry, and we run those ligands into, which we don't know what they are right now. We run them into the mass spectrometer, and that's going to tell us the molecular weight of the active compound. And depending on what machine you're using, it can also show you, it can hint at some structural features so you can start to figure out what the active compound was. So with cannabinoids, we have a whole library of standards that we can use to understand what the active compound was. So that step was probably relatively simple to figure out what the active compounds were. Let's talk about what some of these active compounds were. And it's going to be relatively in order here, but the authors are very clear in saying that the most active compounds in this test were CBGA and CBDA. So you'll also see THCA here, but that wasn't active in some of the other tests they had, just in this step of the discovery process. But it wasn't just CBGA or CBDA. There were some other compounds that had activity, but just lesser activity or less selective activity, so they weren't as desirable. Now let's talk about the most important thing out of all of this, which is the A, the CBGA, CBDA, because you're probably used to seeing things called just CBD or just CBG. And this is incredibly important. So again, we're focusing on this acid component. And this is where we get a little bit into chemistry, and everyone absolutely despises seeing structures because it gives them like memories of organic chemistry and they hate that. But it can be explained a lot easier if we just try to give this a chance and understand this one little component of the chemistry of cannabis. Okay, I'm going to use THC as the example here. So what's most important is that the plant, when it's making these compounds, it's synthesizing these compounds, it is making them in what we call the acidic form. So THCA, this is made by the plant, okay? And when you buy nugs of cannabis from a dispensary, when you're buying that flower, it's going to contain the acidic versions of these molecules because you're buying the plant. You can see that it's physically a plant, so it contains the acidic molecules. What we mean by the acidic molecules is this group right here. This is a functional group on this molecule called a carboxylic acid. You don't need to know that at all, just focus on acid. So this thing in the red is an acid. THCA and most other acidic cannabinoids that are made by the plant are not that active in our body. So for instance, THCA doesn't make you feel high at all, but THC does make you feel high. So when most people are using cannabis, they're going to be looking for the more active form. And that does not contain this group right here that looks like this. We see it's not here, okay? This is the same for CBD. It's not as active in our body, CBGA, etc. It might have some uses, but not usually for what we're looking for. So the way that humans use cannabis as medicine is typically by doing this process called decarboxylation. Decarboxylation because that's a carboxylic acid group and we're removing it. So decarboxylation. Okay. The way that we remove it is by introducing it to heat. So again, we take the flower. This is very relevant when we're talking about flower. We take that flower and we put it in an oven if we're making something like edibles or tinctures. We will take the flower, put it in an oven, heat it up, and it will turn into the active molecule, which does not have that acid. If we are smoking either a joint, a bowl, a bong, a blunt, anything that contains cannabis, and when we introduce that heat, the flower is going to be in that product. So say your flower is in your joint. The flower in the joint is THCA, but when the heat from the fire approaches that flower, it gets converted into THC, not THCA. So when you're sucking in that smoke and that's entering your body, the THC is entering your body, not the THCA. Anytime you're smoking the product, it's going to go through this conversion, making it active in your body. So as I just emphasized too, whether you're making edibles, whether you're making tinctures, or even topicals usually go through this process because the cannabis is heated in an oven, so it's more active in our body. Now let's really emphasize the findings of this paper. The findings of this paper are saying that the acidic versions of CBGA and CBDA are the most active in this test that is testing against the COVID-19 virus. So smoking weed is not going to have the active compounds in it. Smoking weed is not going to help you because the acidic versions of those cannabinoids are being turned into the non-acidic versions. So smoking in any form or most edibles that you're taking are not going to be effective. Tinctures are not going to be effective. If you want the medicine that these authors are talking about, you need to take that cannabis flower and not heat it up at all and extract it in something like alcohol, like high percentage vodka, or something like MCT coconut oil, but never heat it up because that's going to cause it to go from the active form against COVID-19 from the study to the inactive form against COVID-19 in this study. Okay. So a lot of people are like, okay, dude, so you're saying I just chew on the nugs. I would not recommend chewing on nugs. That is not going to be very bioavailable. Your body's not going to be able to use it. It's still going to be trapped inside that nug. You should, if you want to do what these authors are suggesting is the most active, you want to extract it. You want to remove those active compounds from that cannabis flower. Make some sort of extract that you can ingest in a safe way. So something like a tincture or an oil. And then once you make that tincture or oil, if you're making an oil, then you can incorporate that into something a little more tasty for you to use. Okay, but don't heat it up. Now I want to talk about this figure too, because if you're not used to seeing something like this, it's very overwhelming, and it can seem like it's data that's actually generated in the laboratory rather than from a computer. So the data you're seeing here is made using a platform called Autodoc, okay? So this was not done in a laboratory. It was done on a computer. So some people could call that lab work, but we call it in silico work. It's done inside a computer. So there's a few things to break down here. Hold on, I need a quick sip of my tea. Okay. Few things to break down here. First, how is stuff like this even done? So essentially, it is really cool. What they do is they take the receptors of interest and they crystallize it. So in this way, they're crystallizing the S1 subunit of the COVID-19 virus. They're crystallizing that, meaning that they have a crystal structure to see exactly pretty much every atom in that receptor to see how it could potentially interact with the compounds in cannabis. Then there's various different tools we can use online to draw out molecules like they've done here, to draw out the molecules. And then this computer platform is able to, using these algorithms, test hundreds of thousands of different options and ways that that molecule could possibly interact with that receptor. The algorithm's also going to tell you the most favorable way for that compound to interact with that receptor based on just the dynamics of the molecules, because certain portions of the molecules are going to have a higher affinity for certain portions of the receptor. And it's important to note that that receptor is just made up of atoms as well, just kind of strung together in these different orders. So it's kind of like if you think of magnets being near each other, certain parts of each of those are going to be drawn to each other, and that can increase the probability of something interacting in a specific way. But what's really important to note here is that this is done on a computer, okay? So these are just predictions. All we're doing here is predicting how this molecule and this this target protein are going to interact. So something that was really cool that I found from this portion of the publication is the authors are saying that the CBGA binds to the spike protein S1 subunit in a slightly different way than CBDA. So these were the two most active compounds in the study, the acidic cannabinoids. Because they interact with a spike protein in slightly different locations, this could help prevent future resistance. This is just a hypothesis, and future mutations. Because the more ways that these molecules can interact with the spike protein, the more it's going to disrupt its ability to interact with our cells. So that's really cool. It's very promising. And it's also saying that you shouldn't just use CBGA, you shouldn't just use CBDA, you should try to use them in combination. And the authors, I really, really like this about this paper. The authors emphasized that you should be using like full spectrum plant extracts, not just pure compounds here, because pure compounds, I showed that list of all these different compounds that had some activity in these assays. Even if they weren't the most active, they could still contribute just a little bit to the potential benefits of what this paper is saying. So they're saying to use, if you want to utilize what they're saying to use like the full plant extract with all these different cannabinoids present so they can all interact with the spike protein. Now, they also were showing some cell work, and this is going to be really hard to see because it's hard for me to see, and I kind of know what I'm looking for. So this is why I hate publishing this kind of cell work. There's a couple things. First, let me explain what this is saying. They're showing that the cells were stained with an antibody. You can see this in red. It's right here where my mouse is, but it's really hard to visualize. And this red antibody allows you to visualize the replication sites showed that are formed during an infection. So we see that these cells were treated with CBDA and CBGA, the most active compounds here, and we don't see any of those little red dots. But in the control that wasn't treated with any of the cannabinoids, we see a bunch of those little red dots. So this is showing that there's a decrease in the replication sites shown during infection or formed during infection. How did I do that twice? Anyway, something I really just dislike, and this is nothing against these authors, it's just about science in general that includes these. When we're including just a small little snippet of a microscope picture, it's not showing the full story, but this is so commonplace in publications that, again, nothing against these authors, it's just something to keep in note, like keep in mind when you're reading this kind of stuff is, they chose to show this square and this square, but the visualization field could be as big as the screen that you're watching this on. It could be massive, and maybe these just tell the story the best. But again, this data is compelling. It's really cool. I'm excited about it. But just something to keep in mind with cell work. Okay, now something else I want to bring up is the dosing required to get these benefits. How much of these cannabinoids would you have to put into your body to get the benefits that they're saying could be possible with using cannabinoids against COVID? They're very honest about this in the publication, which again, I really appreciate them talking about. So let's focus on this a little bit. The concentration needed to block infection by 50%, which is a very normal metric used in these kinds of tests. 50% of viruses is high, but might be clinically achievable. For example, CBDA administered orally to human volunteers at this dose showed greater bioavailability than CBD. So that's saying that the acidic versions of these cannabinoids is showing better bioavailability compared to the non-acidic versions of them. So that is promising, just thinking about maybe we have a lot of data for the non-acidic cannabinoids, so we can possibly expect that that data would be better with the acidic versions of these molecules. So they're saying it's very well tolerated in beagle dogs. CBDA is very well tolerated at 1 milligram per kilogram of body weight. So if you could calculate your body weight online, you could just go from like pounds to kilograms and convert that to 1 milligram of CBDA per kilogram. It's definitely achievable. The question is, if you ingest that much cannabinoids, how much is it actually getting into the cells of interest? And that's a question we would have to answer with animal studies and human studies by dosing people and animals with these cannabinoids, which we know are very safe and well tolerated, and then testing the serum of their blood to see how high we can actually get the active compounds. And remember, our bodies change compounds when they're entered. So just because CBDA is entering our body, it could be metabolized, it could be incorporated into cells because they're relatively fatty molecules. They could be kind of just uptaken by cells and stored in those cells. So there's a lot of things that can happen in the human body that would cause not as much of the active compound to actually reach our cells. But I'm glad the authors talked about that. But just keep in mind, if you're trying to do this at home or something, you're going to need a lot of cannabinoids to actually be effective from the very preliminary results that they're presenting. Some things I want to totally emphasize in this conclusion. CBDA and CBGA were the most active in their tests in preventing infection by SARS-CoV-2. These compounds are abundant in the hemp cannabis plant, which is totally legal in almost every state in the United States because of the 2018 Farm Bill. So don't go to a dispensary and spend hundreds and hundreds of dollars on a bunch of high THC cannabis flower when you can also get these benefits from using hemp cannabis, which is a lot cheaper. So yeah, hemp is legal in almost every state. I want to emphasize this so much. Smoking cannabis in any way, taking edibles, taking tinctures that are available to you at a dispensary or something, this is not going to prevent COVID infections because the compounds are not in the acidic form. They're in the non-acidic form because you changed it when you exposed them to heat. And the edibles that you buy have probably been exposed to heat to activate them to make sure that they were, making you feel something, like making you feel high if they're THC edibles, and even CBD edibles have been activated by heat in some way. Unless your product specifically says like a CBDA edible, it has been decarboxylated and activated, and it no longer is going to help you. Reminder, all of these studies are done in a laboratory in isolated environments. It does not tell the whole story. I've heard of people going through dramatic things like, I will never vaccinate my family because cannabis helps. Make the decision that's best for your family, but please be informed. Do not spread misinformation. Smoking weed will not help you at all protect against the COVID-19 virus, according to this study. Okay, thank you, everyone. Bye. Comment questions below if you have any more. I might make more videos, but I probably won't because this is not my scientific study.